Hello, everyone. For all of you who don't know me, my name is Jonasz Jeremiasz. I'm from the Institute of Medical Genetics and Applied Genomics in Tübingen, Germany. And today I'm going to tell you something about toxic fragments, proteolytic cleavage, and calpains in the pathogenesis of MJD. So as we all know, there are different concepts of pathological mechanisms, molecular pathology in MJD, and one of the concepts is based on the toxic fragment hypothesis. The idea is that the full-length protein, Ataxin-3, is cleaved into smaller fragments, and these fragments are even more toxic than the full-length protein. Several enzymes were associated with the fragmentation. One of these groups are calpains, calcium-activated proteases, which you can find also in the neurons. This is the group of enzymes we are mainly working on. So basically, calpains are cleaving the full-length Ataxin-3 protein, and we have toxic fragments which are accumulating in the neurons. They can have different effects. For example, they can go into the nucleus, form aggregates, but they can also impair different cellular functions, ER functions, mitochondrial functions. And when these things are impaired, you can imagine that, for example, also the calcium homeostasis is impaired. And this is further triggering an overactivation of the calcium-activated calpains. This is forming a kind of vicious circle, and this is finally leading to a neuronal demise. We can target this pathway differently, so we can directly work on breaking the circle by inhibiting pharmacologically calpains or overexpressing the endogenous inhibitor calpastatin. But what we have done in our study was directly targeting genetically one of the calpain isoforms, which we have associated with the cleavage of Ataxin-3, in this case calpain-1. We have tested first something in cell culture, so we have basically overexpressed mutant Ataxin-3 and knocked down calpain-1. We saw first that when we are overexpressing mutant Ataxin-3, we had an overactivation of the calpain system at baseline, and the knockdown of calpain-1 led to a reduction of this overactivation and also to a reduction of the fragmentation of mutant Ataxin-3. We translated it into an animal model, the YAC84Q mouse, and saw basically the same effects. Here at baseline, we had again an overactivation of the calpain system, and we saw that when we knocked out calpain-1, we could rescue this effect. We saw also that the fragmentation of mutant Ataxin-3 was decreased upon the calpain-1 knockout, and we found some other new markers, basically synaptic proteins like SNAP25 and synapsin-2, which were at baseline in the YAC model strongly or excessively cleaved, maybe a kind of further contribution to the molecular pathogenesis of MJD, and upon calpain-1 knocked out, this fragmentation was rescued again. We looked furthermore at some behavioral aspects of the mouse. So we looked at the motor phenotype. Unfortunately, it was worsening, this phenotype. It was a bit unexpected. But when we looked at different characteristics, we saw, for example, that there was a rescue of the body weight loss in the animals and also an improvement of the overall survival in the animals. So despite having this ambiguous effects on the mouse phenotype, we can say as two take-home messages is calpains are at baseline overactivated in MJD, maybe as another interesting molecular marker, and that targeting calpain-1, in our case genetically, was rescuing some of the hallmarks of MJD. So the main bottom line is that calpains constitute a promising therapeutic target in MJD, but have to be further investigated, of course. I want to thank you. I'm concluding with my acknowledgements and want to thank the organizers for giving me the opportunity to present my data here. Thank you.